Hello and welcome to this session dedicated to image processing using a freeware, ImageJ. And in fact, we will use a variant of ImageJ, which is called FIJ, on your image. Yes, this session comes as a follow-up to two practicals that you've had, one dedicated to immunofluorescence staining, where you've been able to stain the sample and detect the proteins of interest, and the one dedicated to image acquisition. And we will be processing those your already own images to try to quantify few parameters out of your images. This training is split in two parts that are tightly intermingled, where we will go through the theory, basic theory about image processing, and then practicals using the software and using your ready own images. So let's get started. So in previous episodes, you've been labeling cells with three different stainings, two immunolabeling and one which is a dye labeling the DNA. So you've been using DAPI to stain the nuclei, and you've been using two antibodies raised against P16 and Ki67. In this lecture, what we will try to do is first detect all the nuclei, and then knowing where the nuclei are, we will try to measure the quantity of the two proteins within those structures. But to do this, we will go through several steps, 5 steps in fact. We will prepare the nuclei for image segmentation and play with the image display, know how to visualize and handle things that will corrupt the image, like, for instance, the noise. We will then try to classify the pixels into two different populations, the one constitutive of the signal and the one constitutive of the background. So we will partition those intensities into two classes. Of course, if you review your images, you will see that in some places nuclei are quite close one from the other. You've got touching nuclei, and we will try to handle this problem to isolate each individual nucleus. And for this, we will use morpho-mathematical operations to separate the objects. Once we have differentiated the two populations of pixels, it will be time to group the pixels into objects, individualize the nuclei, and get their outlines. And to do so, we will tag the object pixels and constitute what is called an object map to extract at least the object boundaries. Once we have all the object boundaries, this will be time to quantify the signals cell per cell, using those boundaries to extract the associated signal. But before we get started, in fact, we've got to ask this question: what is an image? Well, an image is a representation of a scene. So this is the image is constituted through optics, through a detector, meaning that an image is not the object. This is a representation of the object, same as for this Magritte painting, where it's written, this is not a fume pipe. Yeah, it's not. This is a painting presenting the pipe. So keep that in mind, because depending on the optics that you're using, depending on the system you're using, you will have limitations. You will have corruption of your perception of the object. So an image is not the reality. This is something that is as close as possible to reality, but this is not reality. This is a representation that might be corrupted by a lot of things, and amongst which there is one important thing, which is noise. And we will see later how to process images and try to lower the contribution of the noise. So you may have experienced two types of images. On one side, you may have experienced what is called raster images. Raster images is everyday images. These are the pictures that you take with your smartphone. This is the kind of signal that you're collecting through a webcam. This is an element that is composed of smaller squares, smaller elements, which are picture elements, or as we call them, pixels. This, of course, if you zoom on this kind of image, as you're zooming in, you will progressively see the structure of the image. You will see that the image is composed of a grid, and there is no way you can increase the resolution above the resolution that you got when acquiring the signal. On the other hand, you may have already crossed vector images. Vector images, as depicted here on the right side, are images that are using specific descriptors. These are anchor points, and between points you may have line, and the line may be straight or may have a certain angle. And basically, a vector image is just a set of coordinates, of instructions, or vectors to link those anchor points, meaning that as you've got a mathematical representation of your image, you can zoom in, zoom out, without having the problem of seeing a pattern, because everything is well described. Unfortunately, this is not the kind of images that we are accessing to when dealing with microscopy. So we are dealing with raster images that might be considered as this kind of stuff here. You've got an image on the top left, and you've got here on the side, you've got a grid. This could be an Excel spreadsheet as well, where each block, each cell of this grid contains a value, an intensity. So if the intensity is high, then this is rather light tones, as compared to here dark tones that you may have on the center of the microscope, which are usually lower values. But the question might be, which value are we picking? So first, those values correspond to an amount of light that has reached the detector. So the detector that you're using, whether it's a camera or a photomultiplier tube or whatever, it will convert the light into a signal that then will be transferred to the computer, and the computer will place this intensity over a certain position of the image, over a certain pixel. But of course, there are plenty of possible values in real life. You don't see any sharp gradient of light. You see a continuous variation of the light when looking at the scene. Unfortunately, to represent this the proper way, you would need a lot of possible values. And unfortunately, computers are limited. You can't work with all the value of the universe. So we must quantify the signal that is captured by the detector and scale it, put it on a scale. Use a scale to measure the amount of light and represent the intensities that are collected. And in this matter, especially in microscopy, we are using mainly two types of intensities, of values. We are using this kind of scale, 8 bits scale, and also this one, 16 bits scales. On the 8-bit scale, you have different possible levels. In fact, you've got in total 256 possible values. As the values are starting with 0, so this is a minimum, this corresponds to pure black, the pure white would have a value of 255, which makes a total of 256 levels. If you want to represent a scene more precisely, have a better description of the intensities of your scene, then you would opt for this kind of scale, the 16-bit scale, which has 65,536 different levels. Of course, it will occupy a larger part of your memory as compared to 8-bit, but it will be more precise, especially if you've got a high dynamic of the phenomenon you're trying to quantify. If you have only black and white images, if the signal can be on or off, and that's it, you'd better stick to 8-bit. If you've got really subtle levels, subtle changes and so on, you would opt for 16-bit image. It is important to note that our eye is really bad at quantifying, at comparing stuffs, and at purely quantifying a signal. The human eye is able to distinguish roughly 50 shades of gray, and formulating it that way, it's easier to remember, of course. At the moment, I've been only talking about 8 bits, 16 bits, and you see from the representation that these are gray levels. You would ask, yeah, well, what about color? In fact, the way to handle color images on a computer is we've got two ways. The way you've been using, because you've been acquiring color images, you've been in fact acquiring 3 different images seen through 3 different filters, a red, a green, and a blue, which mimics a bit the way your eye is actually working. So in fact, when talking about color image, some people will talk about 24 bits images because it's 3 times 8 bits. You've got 8 bits for red, so you've got a black and white image and then you apply some kind of overlay of color, red color, one red shade associated to one intensity over the 256 intensities for the red channel. Same goes for the green channel and for the blue channel. And in fact, when talking about color images, there are several ways to think about color images. And in fact, this is a bit confusing, and you will see it in the practicals. You may have color images, and you may have colorized images. A color image is basically made of 3 layers, one red, one green, one blue. But each layer is a black and white image, plus you've got this color coding, so with the shades of red, shades of green, shades of blue. But the 3 information are separated. You could do also same thing by having an image in black and white, and then coding the colors as intensity. Let's say that the first intensity is rather brown. Okay, where do I have brown? Here, maybe here, maybe here. If I look at the values of those pixels, the values will be 1. If I want to know how to color those pixels, I've got to have a look at the dictionary here, which says when you find 1, put brown. You may have blue, for instance, in the mid-range, around 128. And wherever you will find 128, you will put blue. So you see, instead of having 3 times 3 layers, 3 different layers, each one being a bit, you could use what is called an indexed color image, where you have some indications on how to color, and the dictionary of colors associated to it. In terms of memory on the computer, it will be smaller than a pure color image. Talking about the computer and storing an image, you all have been playing with images. You've been maybe playing with some TIFF images, maybe playing with JPEG images. And if you've been using microscopes, you've been using specific image file formats that comes out of the microscopes. So TIFF, in fact, is an acronym for tagged image file format. So a file on your computer, when it's an image, is not only the data of intensities, it's also what is called metadata. For instance, in the file, at the top of the file, you will have information such as the image width, its length, the bit per sample, is it an 8-bit or is it 16-bit, type of compression, a lot of information in this field, image description, which may contain things like the camera you've been using, the model of the camera you've been using, etc., etc. In a TIFF file, everything is well formatted according to some rules that have been set by the Adobe company. And so the metadata are composed of a tag, and then a content. JPEG. JPEG stands for Joint Photographic Expert Group. In fact, this file format was released when we had the first numeric cameras. The problem is the detectors were so big that the files, the information that they will give back, the information was so big that it couldn't fit on the drives that were available at that time. So what this expert group has done is try to find a way to shrink the size of the data so that it can fit on the storage media that were available at that time. They did it in a way that will impair the original signal that you collect from the detector, based on some lack of the human eye. A JPEG picture will, how could I say? It will remove part of the details present on the image, so that it's easier then to compress and make it fit on a storage medium. So I've been talking about metadata, which is a part quite important of the file. And of course, the file contains the information relating to the image. It may store the image data raw, simply by putting side by side all the intensities, or it may use what is called a codec, so it's an algorithm dedicated to compression and decompression in order to try to shrink the content. So you've got several algorithms, run length encoding, for instance, the LZW, which is also used in zip files. JPEG is actually, it's not, people use JPEG as a file format, the real file format is JFIF, and JPEG stands for the compression that is used within this kind of file. But nowadays, everyone is talking about JPEG. So JPEG is a compression that is said non-conservative, meaning that you will lose part of the details over the image, whereas RLE or LZW or algorithm that will keep the full information over the image. And on the next slide, I've got examples to show you basic concepts about how compression works. Let's assume that we've got this sentence, AAA, BBB, AA, CC, etc., etc. You see that you've got a lot of repetitions of the same characters, and the repetitions take place directly in the row. So instead of stating each single character, it would be easier to have the number of repeat plus the character. And you see that by doing this, the sentence gets really shortened. And so coming from the top and going to the compressed sentence, you may now go back without losing any information. It's easy, so you've been compressing and you haven't been losing any kind of information, which is important. This is why this kind of compression can be used when you want to do image analysis, whereas JPEG is always a bit touchy because you're losing part of the details and part of the information. The other way to create a compression algorithm is to have a sentence like this and look carefully at this sentence and realize that there are segments of this sentence that are repeated. The one in red here, ABC, the one in cyan, EF, they are repeated. So the thing we could do is try to shorten those sequences by associating one sequence like ABC to a number, let's say 1. So 1 would be used instead of ABC within the full sentence. Now that you've built a dictionary, you can re-encode the full sentence, but using only those numbers. Of course, in reality, these are not numbers, otherwise you would limit yourself to 10 different words within your dictionary, and this wouldn't be efficient. And once more, of course, whereas this first compression was directly efficient, in order to have a real impact on the size of the file on your computer, you would need to have a longer message and to build appropriate dictionary. This is the kind of thing that is actually used in zip compression. Now I think you know a bit more about the what is an image, and it is time to switch to actual image processing using ImageJ. So I'm sure that you've already installed Fiji software. It is here opened. And I also have a folder where I've got all of your images. So to open images with Fiji, it's quite easy because you only have to drag and drop one or more images to the toolbar. Before we start, I've got to show you the interface of Fiji. It may be different on your side, especially if you're using Windows, because all the menus that I've got on the top are for Windows users. They are docked on the top of the toolbar. Here on this Fiji toolbar, you've got tools, and you've got here what is called the status bar. You see, when I go over each tool, I've got a small description of the tool. This is a way for the software to communicate with us. So if you have to have a look at informations, you will find them here. Most of the time, you will find them here. Now, to open an image, in fact, I won't open only one image. I will take the 3 images of the series, so DAPI, KI67, and P16. I select the 3 images, I drag them onto the toolbar, and you see that the status bar now states drag and drop. It means that it has detected that you're currently trying to open some images. I will release, and it will open the 3 images. So by default, it will open the 3 images in what is called a cascade. So one on top of the other. So you would directly go for clicking on the window, displacing the window, maybe using the magnifying glass that you've got here, or the plus or minus key on your keyboard to zoom in and zoom out and place the 3 images side by side. But refrain from doing that, because there is a function to do it automatically. You would have to go to Window and then to Tile, and it will put all the images side by side and adapt the zoom. If by mistake your ImageJ toolbar is hidden by one of the images or more than one of the images, what you will have to do is click on one of the images to activate it, and then press the return key. If you do this, the ImageJ toolbar will pop up. Okay, now let's have a look at one of the image. Here you also have some kind of status bar, but dedicated to the current image. Here you would have the size of your image expressed in pixels, if you don't have any calibration, spatial calibration over your image. And then you would have the type of the image. And you see that this is RGB. RGB, 3 layers. This is a color image. And indeed, the 3 images are RGB. They are color images. So if you already have 3 layers, how can you merge them to have a new color image? Well, with ImageJ, you can't directly. There is a small operation that we will have to do. To confirm that you've got a color image composed of 3 layers, red, green, blue, you may move the cursor over one of the image, over some of the pixels, and what you will see in the status bar here, you will see first the XY coordinates of the pixel, plus 3 values, one corresponding to the quantity of red, the other one of green, and the third one of blue. By the way, you see that even if it's the nuclei appear as blue, this is not pure blue, because you don't have a 0 value in the red and green. You also have signal in the red channel and in the green channel. So if we want to overlay the 3 channels in order to see the relative position of the 3 markers of interest, you would have to go to Image, Color, and Merge Channels. Unfortunately, this won't work on color images. What we will have to do first is to go to Image, Type, and change the type of the image. It will basically do an average of the red, green, and blue signal and put it on a new image. So 8-bit. You see that now the image is black and white. Now it's stating 8-bit. And now if I go over some of the pixels, you will have only XY plus only one value. I will repeat the process for the 2 other images. So image, type, 8-bit. The signal seems quite dim, but if I go over the image, I see that not all the values are equal to 0, and most of the values are there, but they are really low. And as low values are displayed as black or dark gray, this is normal that the image seems to be all black. Once more, don't trust your eyes, because if you don't have many contrasts on the image, you won't be able to see anything. I go to the green channel, image, type 8-bit. Okay, now that we've got the 3 channels, we can overlay them. And to overlay the 3 channels, we would go to Image, Color, and Merge Channels. Now we will have to make some choices. Choices about the colors in which we want to display the 3 channels. But don't worry, this choice that we make here, we can later on change it. So red, green, and blue. So the obvious choice here would be to set the DAPI in blue and the other channels in green and red, depending on how you've seen it on the microscope. But there are a cleverer way to assign a color to each channel. You've got to know that a human eye is really highly sensitive to green, a bit less to red, and in the worst case, the worst case is blue. So here, the information carried by the DAPI image is just to position the nuclei. So we don't need to have a precise view at the nucleus. We just need to know that they are there. Maybe there are more details on that signal and that signal. In that case, if, for instance, this channel is the one where you have a lot of details, you would put it in green. And then the other one, you would put it in red. So this is how to make choices about colors, not based on what you've seen on the microscope, but on what you want to show on your image. You also see that you've got 4 additional possible colors: gray, cyan, magenta, and yellow. Cyan, magenta, and yellow are things that are used especially for colorblind people. It will be easier to see cyan, magenta, and yellow as compared to pure blue, red, and green, especially if you're lacking the detectors in your eye for one of the channels. So, for instance, I could go for cyan for the DAPI, and maybe magenta for, let's say, P16, and finally the last channel to yellow. I want to create a composite, because if I don't create a composite, then I will end up with an RGB image, which is a color image with 3 channels on which I will struggle to do additional processing. So keeping it as a composite is better. But be warned that composite is something that is specific to ImageJ. For instance, if you reopen a composite within Photoshop, you won't see the 3 layers. And before reopening it into Photoshop, or maybe re-importing it into PowerPoint, you would have to go to Image, type, and convert to RGB. We don't need to keep source images, and we don't need to ignore source lookup tables. So tables for correspondence between intensities and color, because we don't have any color on the 3 images. So now I click on OK, and I've got the 3 channels. The thing you would like to do is have a look at here, the status bar. You see that it's colored. You may also notice that the outline is colored. This is channel number 1 out of 3. And you've got a slider down there to navigate from one channel to another. It won't change anything on the display of your image. It will just change the look here. Each time I activate a different channel, I've got some information about on which channel I'm working, and also what kind of color is associated to this channel. Of course, right now I see the three channels. Even if the yellow channel is quite dim, I see the three channels at once. If I want to play a bit with the display about who is displayed, I could go to Image, Color, and Channels tool. Here, in this box, I've got a drop-down list to select the display mode and tick boxes to enable or disable channels. For instance, if I want to disable the first channel, which is the DAPI channel, I just have to untick it. And now the thing that I see is only an overlay of channel 2 and channel 3. With the drop-down list, I could decide to see only one channel at a time with its color. So here, channel number 2, channel number 3, which is really dim, and channel number 1, which is the nuclei. I could also select to see the information in gray levels rather than in color by selecting the grayscale option. I will go back to color just to show you that you've got this More button, More menu. If you click on it, you have plenty of additional operations, but then you see here some basic colors. For instance, if I want to recolor this channel in blue, I could select blue. I could go to the second channel and switch magenta to red, and switch yellow to green. And now if I go back to composite, I will see the 3 layers overlaid. Here it is. Of course, I know that there is signal in the third channel, but I can't see it. So how could I see it? How could I make it visible? This is exactly what we will be looking at with a bit of theory first. So we've been doing the 8-bit conversion, we've been overlaying, playing with the channel tools, but now how to play with the display. So this is the start of the image processing, trying to work on the display. And before we do that, we must have a look at what is an image. An image is a collection of many things, a collection of pixels, a collection of frequencies, and a collection of objects. For now, we will have a look at the image as a collection of pixels. We may consider the pixels as independent or the pixels as part of a certain neighborhood. For now, we will have a look at the pixels as, consider the pixels as independent. From there, we can start to do some image processing and in particular do some histogram manipulation. But first, what is a histogram? The histogram, when talking about the image histogram, is a simple representation where you've got in the x-axis all the possible intensities found on the image, and on the y-axis, you've got the number of pixels carrying that precise intensity. On ImageJ, you will see, especially in one box, that on top of the histogram, you've got an additional representation, an additional line which is displayed. and we will talk about it a bit later. So let's go back to Fiji, and let's see if we can generate this histogram. A histogram, so you can manipulate the histogram, and in this case it will be already a processing. But a histogram is also a first analysis over your image. You're analyzing the content of your image. So it's quite normal or quite okay to find the function in analyze histogram. Here it's asking if you want to include the 3 images in your histogram. Of course, as the 3 channels are correspond to 3 different signals, you just want to focus on one signal at a time. So do you want to include all 3 images? No. Okay. Here you see that you've got an 8-bit image, because the intensities are starting at 0 and ending at 255. And in the case of this specific signal, the minimum intensity is 31 and the maximum intensity is 154. You see that you've got maybe 2 peaks that are close one from the other, or maybe 1 peak and then 1 bump. I don't know. And we will try to investigate it. So that maybe the thing we will do first is press the live button. Live button will allow to update the histogram depending on what we are doing on the image. Like, for instance, switching from channel 1 to channel 2. On channel 2, what you see is a shape which is really different, still over the 0 to 255 range. The minimum is 3, the maximum is 149, and you see that associated to this gradient of intensity, of all these shades of gray, you've associated a certain color, a certain shade of red. This is how it works. This is how lookup tables work. They associate color to individual gray levels. On this histogram, what you see is a first peak, and then a large but lower bump. Lower in terms of number of pixels, but higher in terms of intensity. So the question is, what is this big peak? And why are so many pixels in this class of intensity as compared to the lower number of pixels associated to higher intensity? The best way to investigate is to have a look at the image and to see that a lot of the area of the image is black, is composed by the background. And you've got maybe 1/3 or even less of the area of your image that accommodates signal. So it could be that this big peak is the background and this lower bump is the signal. And to check that, what we will do is use the selection tool here, the rectangular selection tool, and draw a rectangle over the background. If we do that, as we are in live mode on the histogram, we will have the histogram only on this part of the image. And if I click outside, the region of interest will disappear. And what you see, I had a tiny little selection, so this is why I got really tiny histogram. So if I click outside, I don't have selection anymore. I see the peak, the bump. If I select the background, I see only the peak. which confirms that the peak corresponds to background. Now, if I draw over the signal, I see a bump that is scaled now. To confirm that this is signal and outside its background, the thing I will do is click inside the region of interest, let it click, and move the region over the background. And in the meantime, I've been switching the channel. So signal, background, signal and background. And if I click outside, I've got the full histogram over my image. So this will be useful for one of the next steps when we will try to differentiate the signal pixels from the background pixels. And there is surely a way to set a limit in between the two and consider only those pixels, the ones with a higher intensity. And as I've got a peak and a bump, I may be able to automatically find a value that will split this intensity domain into two. So we've seen that the histogram may give some information about pixels that are belonging to the background and pixels that belong to the signal. But this is a good thing because we are progressing, and especially for the future analysis of the images. But at the same time, it's not giving us any improvement over the display of the image. To improve the display, we will have to use different tools. Those tools, you can find them in here, in Image, Adjust, Brightness and Contrast. When using this box, and using any box within ImageJ, be sure that before launching the box or using the box, you've been clicking on the image on which you want to work. These operations, those processes are done only on the current image, on the active image. Here on this box, you see that you've got a minimum value, a maximum value, and a display of the histogram. And you've got a line here. Just imagine that this range, those shades of red are here. It would mean that 0 will be displayed with black. 255 will be displayed with pure red, the red that is here. Unfortunately, the information is only occupying half of the range, meaning that the brightest red that I could have is a dark red here. So the idea now would be to indicate to ImageJ that the maximum to be displayed is not 255. The maximum to be displayed is 149. So by using the cursor, the maximum cursor, I will move to the left and you will see it will become, the signal will become brighter and brighter. And if you have a look here, you will see that the display is updated as well. So here I would go for 149. Same goes for the 0, which is not 0 but 3. It doesn't make much of a difference, but we can move it to 3. And if you struggle putting it at 3, you can always use the set button and manually enter the value to have a minimum displayed value at 3 and maximum displayed value at 149. Now, the thing we could do is do exactly the same for the other channels. Here, I will move to the green channel that I can't see on the screen right now. This is because the signal is encompassed in a range of 0 to 27. And so, meaning that you've got only 1/10 of the full range that is used, meaning that all the values will have really dark shades. So now I could go back to brightness and contrast. And first I click on composite, then brightness and contrast. I see the histogram. I can use the set button and 0 to 27. And you see that it makes a huge change in the display of the image. As for the DAPI, well, I don't think that there is much to do, but a bit of improvement can be done. So minimum is 31, so set 31 to 154. You may ask, what happens if I lower the maximum? Well, if I lower the maximum, this value, 70, is the new bright blue. And after this bright blue, there is nothing left. There is no even brighter blues. So all the intensities from 70 on, they will appear with the maximum value. And this will generate something that is called saturation over your image. Of course, this saturation is only on the display. You've been acquiring the images without saturating, without reaching the top of the range. So this is only a matter of display. Okay. So basically, you will set the minimum value and define what you call black. You will set the maximum value and define what you call bright blue. And in between, the intensities will be distributed linearly over the blue shades. So this is what is exemplified here. In fact, you've got the regular histogram displayed in brightness and contrast with all the recorded intensities and the count of pixels carrying each individual intensity. And at the same time, you've got this line which reflects the way you associate the recorded intensities to the displayed intensities. If you move the minimum toward the right, you will end up with values that can't be displayed because you don't have any displayable intensities for them, so they will automatically appear with the first intensity, the zero intensity, or the shade corresponding to the zero intensity. And to the opposite is also true. If you lower the maximum, all intensities above will be displayed with the maximum of the color range that you've selected. Okay. So we've seen that an image is a collection of pixels, which is not new. And now we will see that pixels, they do have a certain neighborhood from which we can benefit to do some additional processing, such as the spatial filtering. So why should we do some spatial filtering? Let's go back to Fiji. Let's activate the first channel, which is DAPI, and let's duplicate this channel. So we will use this command that you will find under image, duplicate. You have a box in which you can define a new name for your image. You can select to duplicate the hyperstack because this container with 3 different layers is called an hyperstack. I don't want to duplicate everyone. I just want to duplicate channel number 1. So I click on OK, and now I've got a copy of the DAPI. If we have a precise look at nuclei, so I will focus on this nucleus and zoom on this specific nucleus. To do that, I will just point the nucleus and press the plus key. Then point, plus, etc. And basically the zoom is centered over the point at which on which I've got the cursor. If you look at this nucleus, you will realize that there is some kind of texture over the nucleus. And if you want to precisely see the texture, you will have to use the tools here, and especially the line tool. Draw a line across the nucleus and do some kind of analysis. Let's move it a bit, maybe like that, so that it goes over several nuclei. Before doing the analysis, we will go to Analyze, Plot Profile. Here you've got a graph that is presenting the intensities collected over the line. So here you've got all pixels encountered along the line. They are here. And here you've got the intensity that are collected along the line. And what you will see is background, nucleus, maybe another one, and then another one. If I press the live button, then the graph will respond to any modification I will do on the line. So what you see here is within this nucleus, I don't have an intensity that is homogeneous. Between this last nucleus and the next one, you see that this part of the curve is not going back to the same level as the background I had here. You see that this nucleus here, it has a higher intensity, but this is a variable intensity. So let's imagine that I want to separate the background pixels from the pixels constitutive of the nuclei. The thing I could do is set a certain limit, a certain minimum intensity that you should have to be considered as a pixel belonging to nuclei. Let's imagine that I set the intensity here. What will happen? I've got a connection between this nucleus, that one, and that one. So all this, meaning all this, will be considered as a unique entity, which is not what I want to do, because I want to have measurements done nucleus per nucleus. So you would raise surely a bit the minimum intensity to be considered as pixel belonging to nuclei. It would be okay for that one, which would be separated from this blob. So containing this, this, and this, so 3 nuclei, and it would be separated from the other nucleus. So to separate that one, and that one, you would increase the limit to something like that. But of course, by doing this, you see that you don't take the full shape of your nucleus. You're losing part of the signal here. So maybe this is not a good idea to try to raise the limit. Maybe we should first do something to try to homogenize somehow the signal to make it easier to distinguish from the background. So to do that, we will have to use some filters, and I will explain you how filters work in the next part of the tutorial. So let's see what we call spatial filtering. As I told you, we are considering a pixel within a certain neighborhood. So for instance, on this example, we've got the original image. We will focus on only one pixel for now, and we will focus on a neighborhood of 3 by 3 pixels, on a square of 3 by 3 pixels. To do filtering, what you will need is both the original image and the filter. The filter is simply a small matrix, a small grid of 3 by 3 pixels, if this is a neighborhood that you want to play with. In this matrix, in this filter, you will have coefficients. At the moment, you've got 1 everywhere. Now, you will need a destination image on which you will put the result of some processing between the original image and the filter in the specific location you're filtering. So I want to filter this pixel. I will take all the intensities within the neighborhood, and I will modulate those intensities with the filter in order to compute a new value for this pixel. The thing I will do is take the intensity, multiply it by the first coefficient, second intensity, second coefficient, third intensity, third coefficient, and so on and so forth. So 168 times 1 plus 151 times 1, etc., etc. I end up with a sum that is larger, really larger than the original pixel intensity. So this is why I will normalize this result by dividing by the sum of all the coefficients I've got within the filter grid. I will end up with a number with decimals, but decimals can't be stored into an 8-bit image where you only have integers. This is why you will drop everything that is after the dot. The new intensity, the new computed intensity is 180, and I will put it on the destination image. By doing this process, in fact, you've been summing all the intensities and dividing by the total number of intensities, 9. You've been, in fact, computing the arithmetic, the average over your neighborhood. This is why this filter, this precise filter, is called a mean filter, an averaging filter. The mean filter is not the only one that exists, because depending on the coefficient that you will put within your filter, you may end up modifying the image and enhancing certain details within the image. For instance, if you're using the Zobel filter, putting 0 vertically and putting equivalent coefficients on one side and on the other side of the zero line with different signs on both sides, you will enhance the vertical lines on the image. If you tilt this matrix, then these are the horizontal lines that you will enhance. And if you sum the square of the two results, then you would have enhanced all the outlines of your objects. So the first type of filters I've been talking about are called the linear filters, because you have a matrix and you apply the same matrix to all pixels over the image. And this is exactly the same processing that is done on each single image. There is a second class of filters that you could use. These are called the rank filters, where instead of applying coefficients, what you will only consider is a neighborhood. You will collect all the pixels within the neighborhood, and you will rank them from the lowest value to the highest value. And then what you will put on the destination image is dependent upon the kind of filter you want to use. You may use a minimum filter, retaining only the minimum value, a maximum filter, only retaining the maximum value, or a median filter, which will pick the value that is in the middle. And in general, on the images from microscope, the median value corresponds to the most represented value in the neighborhood. So if you've got really dark spots or really bright spots and you want to homogenize the intensities over a certain neighborhood, median might be the most convenient filter. Now, the thing we will do, we will go back to Fiji and we will play with the two types of filters. We will focus on two specific filters, the mean filter and the median filter. Okay. Back to this image where I've got a line, and on the side I've got a graph, the graph corresponding to the profile plot over the line, and this is still in the live mode. I click on the image to make sure this is this image that is active. This graph is also an image. So if I do a processing, it will process the graph if the graph is active. This is why I make sure to always click on the image I want to process before going to the menus. In this case, I will go to the menu called Process, and I've got filters here. And for you to see better the effect of the filters, I will replace the color table I've got on the image. From blue, I will switch it to gray levels. It will be easier to see. To do that, I could go to the Channels tools, but I would go to Image, Look at Tables, and Grays. Okay. I click on the image, and now I will play with the filters. So Process, Filters, Median, Mean. I will start with the mean filter. You see that in this box, the neighborhood is not based on the square, but rather on a circle. This is why you're asked to enter a radius. So let's try with a radius of 2. Don't click on OK, because you've got a preview button that will give you a preview of what will be done over the image. So I tick the preview box, and I don't see much over the image. But on the graph, I see that the little peaks there are smoothened. I could increase the neighborhood, let's say to 5. And you see that the image becomes a bit blurry. It's more homogeneous within the nuclei. And I have the bumps, and I will be able to separate a bit more, a bit easy, in an easier way, the nuclei. If I go to a really, let's say, large radius, of course, I will lose the outlines. And you see that the outlines are spread around, meaning that I won't have a sharp edge around the nucleus, which could be an issue when trying to indeed isolate the outlines. You see, we've been spreading and in fact losing the sharpness of the outlines. So, well, it could be a way to smoothen the profile, to homogenize intensities, but you've got to be careful about the neighborhood. Let's cancel and let's try with the median filter. You see that it's taking a bit more time for the computer to get the result. I don't see much of a difference right now, but if I go to, let's say, 7, you see that it will homogenize really a lot locally the intensities, but look at the edges of the nuclei. The edge, the rim of the nucleus is really well defined. So even if we are increasing the neighborhood, I won't have the same spreading effect as with the mean filter. So 25 was maybe a bit too big, but let's try it. You see the progression bar here. Okay, so we are starting to lose a bit the shape of the nuclei, but you can't say it's not homogenized. So let's be reasonable and go back, let's say, to 7. And I will click on OK. And when clicking on OK, the new intensity values will be pushed to the image. I can unzoom to see how it looks. And the thing I can do is put the two images side by side, clicking on the original image and using the channels tool, moving to the grayscale display to see before and after processing. I could zoom on one image, zoom on the other image, and you see that we've lost the texture here, which will make it easier to segment the image afterwards. So next step will be to differentiate the intensities constitutive of the background from the intensities constitutive of the nuclei. Let's have a look at how we can do it, going back to the presentation. We will go back to something that we already know. So considering the image as a collection of pixels, of independent pixels, only considering their intensities, and having a look at the histogram of the image. As I told you before, Having a look at the histogram, we've been able to differentiate two populations of intensities. The ones that were constitutive of the background, lower intensities, and a high number of pixels composing this set, and another class of intensities a bit higher that are constitutive of the signal. So now the question is, how do I find the limit between the two? Well, I could try to fix it manually, for instance, here, or I could try to use some automated methods. For instance, I could try to identify the two peaks, and from the two peaks, select a value that would be at halfway. This is one way to proceed. In this specific example, it wouldn't be appropriate, but this is one way to proceed. Sometimes it works. It all depends on the nature of the information carried by the image. I could consider that half of the pixels are background, half of the pixels are signal, and I will end up with a different threshold. Or I could use the overall image mean intensity as a threshold. These are methods. They don't apply all the time, but they are worth trying. Now, let's see in practice how we can do it. So let's go back to Fiji and try to set an appropriate threshold. So we will do it on the image that we've been processing, filtering. I will unzoom to have a global view at my image. And now I will use a tool that you will find in Image, Adjust, Threshold. By default, it has been trying to compute a threshold, and the result is 60. You see that you've got two values. You've got 60 and 255. And in between the two, you've got this big red box. The big red box delineates the range of intensities that are displayed in red in this overlay. So you see that 60 is not appropriate. I can manually change the value, and if the value is too high, I will miss some of the nuclei. If it's too low, I will have some nuclei fused one with the other. It may happen that the red area is encompassing only the background. This is due to the fact that you have been unticking this box. This box ensures that you're considering white pixels over a black or dark background. So 71 is not bad. Instead of doing it manually, you may want to try to use automated methods. And there are plenty, each one relevant to one use case. If I well remember, this method, the triangle method, is working quite okayish on this image. So I will stick with that, and with that we have defined the minimum value a pixel should have to be considered as part of an object. If we want to fix this value, if we want to now get a new image where we've got only pixels from the background and pixels from the objects, then we could use the Apply button. I will press Apply, and now I've got a black and white image. And if you're looking at the intensities in the status bar from Fiji, you will see that background pixels are tagged with a 0 intensity and object pixels will be tagged with an intensity of 255. Okay, so we've been adjusting the threshold, and we've been creating what is called a mask. The mask is when you start with an original image like that one, you decide of a limit intensity, and then any intensity below becomes white and above becomes black, or the opposite way around in our case. In our case, the above the limit intensity, it will become white, below it will become black. You've just generated what is called a mask. And on a mask, you can do plenty of operations, which are called morphological mathematic operations. And in particular, I will show you one and explain to you one specific operation, which is called Watershed, and which will be really useful, especially knowing the issues that we will have with some of the nuclei touching. If you focus on this set of nuclei, you will see that you have a continuity between this nucleus, and that nucleus. So we must find a way, using image processing, to split them. We've been doing our best with the filters, our best with the threshold, but still we have some connections here. We will try to get rid of those connections using specific operations that apply to masks. So this specific operation is called the Watershed transform. And to apply a Watershed transform to a mask, the first thing you need to generate is what is called an Euclidean distance map. Basically, each single pixel from the object will be tagged with an intensity corresponding to the distance, to the shortest distance to the background. So all pixels on the outline of the mask for an object, they will be tagged with an intensity of 1. All the pixels on the other ring around the object will have an intensity of 2, etc., etc. If you color code those distances, this is what you will get. You see a concentric distribution of the intensities corresponding to the distance to the background, and you will also see that you've got here and here the pixels that are the furthest away from the background. You could represent it as a geodesic map. So with the peanut like that, you will have the pixels close to the background, then the pixels a bit further away, and the furthest away from the background, and then the opposite way around. So you will have, let's say, two mountains side by side. If you flip the mountain, it will go down like that and up. And this is exactly what is shown on this diagram, which is a simple profile along a line which crosses the peanut. Here, the two spots corresponds to the maximum, the two peaks, the two points that are the furthest away from the background. Let's imagine that now this shape, you printed it in 3D. You've made holes on the two tops, on the two sides, here and here, and now you will add some water from the outside. The water will find the first peak and try to get into the first mountain here. If you add water, the water will jump in the first basin and in the second basin. And if you repeat the operation, at some point, the two basins that are separated here will merge. But before it's merging, you will put a barrier in between the two, a barrier that is called a watershed. And by reporting the position of this barrier on the image, you may be able to separate the two basins that you were able to see here and here. So now the idea will be to do exactly this kind of the two basins are here and here. So it's quite normal to find a watershed in between the two here. So next step will be to use the watershed operation that I've just explained, and we will use it, of course, with Fiji. This kind of thing only works if you've got a binary image. So you should have applied the threshold before trying to launch this operation. I will do a bit of cleanup because threshold, I don't need it anymore. This graph, I don't need it anymore. The histogram, I don't need it anymore. I only need to have, let's say, side by side the original image. Maybe I could even switch to the composite mode on the original image. So this was an error message saying you don't have an histogram window anymore. So if you get it, just close the window. This is okay. And so here is the DAPI mask. On the DAPI mask, I will go on Process, Binary, and Watershed. And now if I zoom on the three nuclei that I was pointing earlier, you see that it has been putting a watershed here and here. The only problem is this nucleus. This nucleus here, it has a hole. And with all the bumps on the side of the hole, of course, it has been able to cut it into tiny little pieces. So the thing I will do is revert by going to Edit Undo. Be careful, in Fiji, not all functions work with the undo function. So you'd better duplicate, but I know that Watershed is working with undo. So I will undo. And then I will go back to Process, Binary, and here I've got a specific tool to fill holes. So I will use it, fill holes. And now I can go to Process, Binary, and Watershed. And you see that it's not splitting anymore. Of course, it's splitting that one, but additional processing should be done in order to rearrange a bit the shape and so on, but I don't want to disturb you with too many information. Okay. So now we have blocks of pixels that are separated and that could be considered as nuclei. The question is, how do I know that this is nucleus 1, this is nucleus 2, this is nucleus 3, 4, etc., etc.? To do that, I will need to consider my image in a different way. Maybe in the third way I've been talking about earlier. To do morphometry, we will do some measurements, nucleus per nucleus, And to do that, we should consider the image not as a collection of pixels, not as a collection of frequencies, but as a collection of objects. And the first step in considering the image as a collection of objects is to perform what is called the connexity analysis. And then we will be able to do the morphometry. So what is connexity analysis? Well, in fact, it's not that complicated, and it's depicted on this slide. We first start with the mask, where here you've got black pixels that are belonging to objects and white pixels that are considered as background. The thing I will do is go all over each single pixel, and when I find the first object pixel, I will give it a tag, and this tag is simply an intensity of 1 for the first object. So finding the first object pixel, I attribute to it an intensity of 1. Then I move along all the pixels. These are background pixels, so I won't care about them. Jump to the next line, background. Oh, this is a pixel belonging to an object. I will have a look around. Is this pixel connected to any pixel that already belongs to an object? No. If not, then I will get and fetch a new tag. This will be the tag number 2. So now I will attribute to this pixel the tag number 2. So I've got tag number 1, tag number 2. I continue along the line. So this is background. Oh, object. Does this object pixels have neighbors that already carry a tag? Yes, there is one here. So now, as the two pixels share a side, they are connected, they belong to the same object, so they should share the same tag. So this is why I attribute to this pixel the tag number 1. And we will continue like that, and at the end, we will cover the full image and get what is called a tagged map. However, depending on the software you are using, 2 rules, at least in 2D, 2 rules may apply. The 4 connected rule or the 8 connected rule. In the 4 connected rule, you consider 2 pixels to be connected only by one of its 4 sides. So they should share one of the 4 sides. On the 8 connected situation, you consider the 2 sides, the 4 sides plus the 4 corners, meaning that 2 pixels can be connected through a corner, so in diagonal. This has an impact on how you interpret what an object is, and if 2 close objects are connected or the same object or not. Look at this example. Here, the 2 pixels are connected through one corner, but we are in the 4 connected situation, meaning that it doesn't count, meaning that this will be an object and this will be another object. If you have a look at the 8 connected situation, then sharing a corner is enough to say that the 2 pixels belong to the same object. And then you will have this L-shaped object. This principle can be extended to the 3D case, and instead of having 4 or 8 neighbors, you would have 6 or 26 neighbors. 6 neighbors, meaning that if I'm a voxel is a pixel in 3D. If you're a voxel, you've got 4 in the same plan, and you've got 1 on top and 1 on the bottom, so 6 connected. If you consider all the diagonals, you've got 9 pixels on the top, 9 pixels on the bottom, and the 8 pixels on the same plane, which makes 26. Of course, there is another parameter that we've been working on earlier that could impact if two group of pixels are considered as one or two objects. This parameter is a threshold. If you use a threshold that is a bit too permissive, then this is how the mask will look like. So, of course, those two groups that you could say, well, these are two objects, they won't be separated if the threshold is too low, meaning that you will have on the tag map only one object. To the contrary, if the threshold is too high, you may disconnect some unique objects. And anyway, you will change the shape of the objects. Just imagine measuring the perimeter of this object. The perimeter will surely not be the same depending on the threshold. So this is why threshold is one way to proceed, but it is not perfect, it is not the best, but this is at least one tool amongst others to delineate, to be able to later on delineate objects. Once you have the tagged map, there are some parameters you can extract. For instance, the total intensity, the total quantity of material associated to each object. You simply have to have a look at the tag map and to collect the intensities of the pixels sharing the same tag. And this way you can sum those intensities and have the total amount of protein associated to your structure. You could also isolate the individual pixels that are connected to the background. Those pixels are the outline pixels. So it's quite convenient to isolate the outline of your objects, but it's quite convenient also to measure the perimeter of an object. Finally, by counting the total number of pixels constitutive of each object, you have a measurement of the area of your object. So you see that the tag map, getting the mask, then the tag map is the first step to do some morphometry over your image. Now, you would ask the question, how do we get the tagged map? Well, once more, we will have to switch to Fiji and have a look at how to proceed. To get the tag map, it will be quite easy. So we will use an analysis tool. So we will go to the menu Analyze and Analyze Particles. So in this box, you have plenty of options. I will ask you to untick everything. And let's have a look at the three first lines within the box. First line, the first box, is a size filter. If I cancel here and if I zoom on the image, you see that you've got pieces of DNA that are not really nuclei. There might be micronuclei. There might be pieces of DNA that are found, I don't know why. Yeah. So, but they are not the nuclei we are looking for. So we should try to focus our analysis on real nuclei. And we've got a criterion to differentiate real nuclei from small nuclei. And this criterion is size. So I will go to Analyze Particles, and I will enter a size filter that corresponds to the expected size of my nuclei. So I will consider that a nucleus is constituted of at least 1,000 pixels. And I will ask ImageJ to show me a mask. So it will give me a mask, but filtered based on the size of the object it will find. You've got this second parameter, circularity. Circularity is a ratio between perimeter squared and the area of the object. This circularity is close to 0 for a line. It's 1 for a perfect circle, and for a square, it's 0.75. So you may ask yourself why I did put 1,000 here. This is a first guess, and based on the result I get from the mask, then I will adapt this size. There are some clever way to do it, but here I don't want to show you all the functions within ImageJ, so we will try, guess, and check. Well, if I compare the two, I've lost this little thingy here, but I still have this big one here. So 1,000 was not enough. I will close the mask, click on my mask and filtered, go back to analyze particles, and maybe, let's say, 1,500. Well, it looks better. So now I've got my size filter. Here I realize that even if I've been filtering small objects, I still have half objects, like that one, touching the border of the image. If I go back to analyze, and then analyze particles, I've got an option here to exclude all the objects that are on edges. This is important, especially if you want to have a look at the area, the distribution of areas of your nuclei. If you've got half a nucleus, well, you won't be able to have precise stats from this kind of data. So it's better to exclude on edges if in that case. And you see that it has excluded the lower nucleus that was touching the side, but not the upper one that is a tiny bit away, maybe 1 pixel away from the border. Yeah, 1 pixel away. This is enough. Now, if we go back to analyze particles, we don't need the mask anymore because we've checked that the things that we are extracting are relevant. But I can show you one thing, which is a count mask here. Let's see the count mask. So this is a grayscale image. And if I point at the first object, I've got 142 as an intensity. And this is coherent over the full object. If I point at a second object, 145, etc., etc. You see that even if the display is not well set, I've got a uniform intensity object per object, meaning that it has done its job of tagging each individual object. If you want to see clearly what Fiji is considering as an object, well, you could encode those intensities as colors. And There are some color maps that are really good for this kind of situation. You would find them in Image, Lookup Tables, which is a list of all the color maps you may use. And in this case, you may want to use the glass bay on dark. And you see, you've got one color per object. So now, with this, we've checked that the tagging, the generation of the tagged map was working. We can close this tagged map, go back to this image and start generating data. Let's go to Analyze Particles, and have a look at the different tick boxes that we've got here. We will keep the exclude on edges. We will ask ImageJ to display results, to clear results, to summarize, and to add to manager. Results is in fact a table that you can later on export as Excel table, containing some morphological measurements over each single object on the image. Of course, if you launch several times the Analyze Particles module, sometimes you want to pull all the data in the same table, or you want to keep the tables clean from one launch to another. This is why I ticked Clear Result. Clear Result will clear the table before putting the new results. Summarize is just Analyze Particles will measure some parameters and give you an average of those parameters in a distinct table from the results table. Add to manager. By manager, Fiji means the ROI, the region of interest manager. It will push to a dedicated window the outlines of each single nucleus. Let's click on OK and see what are the results. So, plenty of things. So, count mask, I forgot to remove it from the show option. Summary. In the summary, I've got the name of the image, the number of objects it has found, the total area that those objects occupy, the average size, the percentage of the area of the image occupied by nuclei, and the average intensity of all the objects. Here, we've got the results table with a number of lines equivalent to the number of objects it has found, of nuclei it has found. So you've got the area and the average intensity for each single object. And in the ROI manager, you've got one line per outline of nucleus it has found. You see that at the moment I've got a yellow outline around all the objects. All the objects. This is because the tick box show all is ticked. If I untick it, then you only see the one region of interest that is selected. If you tick show all, you will have yellow outlines around all the objects and a cyan outline around the object that is selected. This is object number 8, and I find all the parameters relating to object number 8 on the line number 8 within the table. So this is the area of this specific object. You may have different values, different parameters within your table. So you may ask, how do I change which parameter to retrieve? It's something that is done by going to Analyze and Set Measurements. Here, you've got tick boxes with all the parameters you may explore. If you want the parameter, just tick perimeter. If you want to fit an ellipse over each object and retrieve the big axis, the small axis, the angle, etc., just fit ellipse. So we've been able to analyze the particles to get the outline of each single nucleus and push it into the ROI manager. You may ask, what next? Next, we'll be trying to measure parameters such as the average intensity of the two signals within the nuclei. And to do that, we will have a look at how to do it in Fiji. And the aim of those measurements is to generate this kind of table in which you will have the number of cells positive for DAPI for P16 and Ki67 in all the experimental conditions that you've been playing with during the practicals. So this is what is expected from you to fill out this specific table. So how can you do that? Well, first you will need to measure over the original image, but in the regions that we've extracted here. And I can activate the composite image. I could untick and retick, and you would see directly on the original image what are the detections that have been done on the DAPI image. And actually, using the channels tool, I could inactivate the two other channels to check that my segmentation is not too bad. Well, it's missing part of some of the nuclei, but well, at least it didn't require the user to outline manually each single nucleus, which is a good thing. The question now is, how do I quantify the signal? I've got the region of interest, I've got the raw data, unprocessed data directly from the microscope, more or less, with a little 8-bit conversion. So the thing I will do is go to the channel, the red channel. Now, on the ROI manager, I will make sure that I don't have any selection. I mean by having a selection, having one region selected like that. Show all doesn't count because show all is just a display, an overlay of all the region of interest without having one selected. Having one selected will give me the cyan outline here. So I will deselect, and then I will use the measure button to measure the intensity of all regions of interest at once. So I have this result table. From this result table, I've got some tools like results, distribution, and from one parameter, such as the average intensity over the red channel within the nuclei, I can generate this distribution. This is not the best distribution. Anyway, I don't have much of data. It has been doing only 5 bins. But from this, I could say, let's have a look at the list. So I've got 15 nuclei, which intensity is between 0 and 30. I've got 14 between 30 and 45. I've got etc., etc. I could consider, for instance, that the first bin corresponds to negative entities, and then I would have to add all those numbers to get the number of positive cells. Of course, this is a quick and dirty way to do it, and I won't recommend to do that. I would recommend to go back to the image, only activate the red channel, and point at what I consider to be negative. For instance, number 2 might be negative, and I see from the status bar of Fiji that the intensity is roughly 40 something. I could have a look also at line number 2 within the table, so 43, and then I would export those data in Excel or in R and count the number of lines where I've got a value that is higher than what I consider to be negative. And this way, I would have the count of positive cells. If I want to keep this result stable, I would have to file rename and give it a name like channel 2. I can save this as well, save as, or I can select everything and copy and paste, for instance, in Excel. If you're copying and pasting into an Excel, which is a French Excel, be careful, you will have to replace the dot by a comma in order to make sure that you've got the proper decimal sign. Now, I will move to the third channel that is not displayed because I unticked it. So this is the third channel, and now the thing I can do is measure. Same, I've got intensities. I can do the distribution. Okay. And now I can review what I consider to be a negative cell, and from there define a threshold and count only the cells that have an intensity that is higher than that threshold. And this will give me the number of positive cells. If I want to have the double positive cells, well, I should put the two tables side by side and see which values are above the threshold in here and above the threshold that I've been defining here on the second in the second table. That's it. I think that we went through all what I wanted to show you during this lecture, but in fact I've got a little additional bonus for you to show you some advanced stuff that Fiji can do. I will give you a really brief and quick demo of one fantastic tool that exists within Fiji that allows you to record some processes and to reapply those processes. So here we go for the demo. I will close everything. All the tables, etc. And I will start a tool that you find in plugins, macros, and record. This is a small window that will record all the actions we will be performing. So you just have to have it there and to forget about it. It will record everything, including your mistakes, so you've got to be careful. If I drag and drop the images, nothing will be recorded. This is why, for once, I will go through File, Open, and then I will look for the images. And in order to simplify the process, for later on, if I want to have a precise knowledge about which image is which, it's better to have some naming conventions, and I will go to image rename to do that. So let's rename it DAPI. I will open the second image and rename it the third image and rename it. You see that it has been recording the fact that I did open the first image, rename, open, rename, open, rename. Then I click on DAPI and it's recording that I'm selecting the DAPI image and transforming it into a bit. Clicking on the second image, transforming it. Oops. And last image and transforming it. So select, transform, select, make line. Ooh, this is a mistake that it has been recording and that I can remove. Select and transform. I will remove this and make sure that my new tool that is selected is a rectangle. I will have less troubles with the rectangle. What I did was image, well, maybe before doing that, I will go back on the DAPI and I will automatically set the gray levels. I will do it also on that image and also on that image. Basically what it does is trying to find the minimum and the maximum displayable values and trying to find the best way to display all the content of the image on the screen. Now, image color merge channels in order to overlay the three images. So red is, let's say, that one, green is, let's say, that one, and blue is that one. I create the composite and OK. Now, from there, I've been working on the blue channel, so I should go to the third channel. And you see that when moving here, it's not recording anything here. So the thing I will have to do is find a way from the menu to jump to the third channel. So there is a way, which is not the best, but image, stack, set channel, 3. We will jump to the third image. So now the third image is active. It has been recording this. And now I can duplicate this image. Image, duplicate. I just want to create the DAPI mask from image number 3. Okay, now I've got the DAPI mask, and you remember that I wanted to smooth the intensities. To do that, I've been using process, filters, median. Radius was 7, okay. Once the filtering was done, the thing I've been doing is adjusting the threshold in order to partition the image into background pixels and object pixels, so threshold. And I've been using this automated way to set the threshold, which is called triangle. Then, when I've been happy with the threshold, I've been applying the threshold in order to get the mask. We've got a mask. From the mask, I had to cut the touching nuclei to make separation using the watershed, but before doing the watershed, I had to fill the holes there. So first, process, binary, fill holes. Second, process, binary, watershed. From there, I've got a mask with nuclei. I've got to get their outlines. So analyze particles. I don't want the count mask. I don't want the results because I will get the results in a different way later on. I just want to exclude everything that is on edges. I want to push the outlines to the ROI manager, and I want to filter the objects based on the size. Now that I've got everything in the ROI manager, I can close the mask. I can activate the composite image. I will have to jump to the first slice to do that, and to get it recorded here, I've got to reuse the trick that I've shown earlier. So image stacks set slice 1. This is channel 1 that is active. I've got the ROI manager with all the region of interest. I can deselect to make sure that I don't have any selection before measuring. And now I've got all the measurements for channel 1. If I want, I can do the distribution. And to be sure that this results table is about channel 1, I will file, rename it. I can then go back to the composite, use the same trick as previously to activate channel number 2. Make sure that in the ROI manager nothing is selected, so deselect before measuring. And now I've got the second table from which I can draw the distribution. And this table too, I will rename it. Oops. Select the table, file, rename, channel 2. So channel 2, channel 1, I've got all the measurements, and now I can process the data and fill the table. So you may ask, why did I record all this? I recorded all this for a simple reason. If I close everything, table, table, remove the close all instruction. If I press create here, I will have what is called the macro editor, where I've got all the instructions. Everything that I've been doing is recorded. This file can be saved. File, save as. This is a small text file that I will save on my desktop. And I could also, I don't need the recorder anymore. I don't need this. I will close the ROI manager. This is a way to empty it. I will close everything. And now I only have to run this. And basically, I end up with the two tables that I've been manually obtaining. If you want to redo exactly the same thing on a different set of images, you will only have to change the path to the files you want to work on. All the rest, as I've been standardizing the names, all the rest is supposed to work. So that's it for the little bonus about macro programming using ImageJ, VG ImageJ. There are plenty of stuff you can do with this software, and I hope that you enjoyed this tutorial and that you will be using it for your report about the practicals. And feel free to ask questions if you've got any. So with that, I'm signing off. So goodbye.